really good examples of applications both to pathogens, to weeds, and now, not to leave anyone out, we want to make sure that the insects get their moment in the sun. And so I'm very excited to be able to introduce Dr. Bob Koch from the Department of Entomology. Bob is an associate professor and extension entomologist and the director of the graduate studies program in the entomology department. His research program focuses on applied ecology and integrated pest management of arthropods associated with soybean. Dr. Koch is a PI on an MIT PPC funded grant on decreasing environmental impacts of soybean aphid management that is supported in part by the Minnesota Soybean Research and Promotion Council. So I'm excited to hear from Bob and his work towards moving soybean aphid scouting from the ground to the air.
Can you guys hear me okay if I use this microphone, or do I need to hook myself up? We're good? Okay. So I guess soybean aphid's a hot topic. Early on, Rob showed you the 4 MIT PPC symposia that they've had, and he's twisted my arm into speaking at 3 of the 4 now. So I'm not sure when he's going to get sick of hearing from me. But like Rob said, my position here, I focus on ecology and integrated pest management of soybean pests, mainly soybean aphid. And through my extension appointment, I get a lot of opportunity to interact with farmers, crop consultants, agronomists, with companies. And around 2013, when I started getting a lot of questions about drones and remote sensing for insect pest management, especially soybean aphid. And I didn't have any answers for people. So, fortunately with my position, I can take those questions that I'm getting from the extension programming, bring it into my research program, employ some, which way are we going here? Oh, the remote, maybe. Okay, sorry, I'm not smart enough to run this thing. Yeah, I can ramble on. I've done lots of field talks, so. Rob, do you? Well, anyhow, Rob's working out that. So I've been able to get a couple graduate students who are really excited about remote sensing technologies and spend a lot of time pestering people like Joe Knight, David Mula, other folks that are a lot smarter in these areas than me, and we've kind of taken it from the point where we have no answers at all to these questions, and I was pretty doubtful that it would really have any utility for soybean pest management, to a point now where I'm kind of reconsidering that and thinking, well, maybe we're onto something here, and looking to hopefully take it to the next levels for broader application.
You're doing good.
Sorry, I broke it.
No, you're doing good. Just keep going.
Do
You want to say a word about the history of soybean aphid and how long it's been here and why it's become such a problem?
Sure. Yeah, so soybean aphid, it's originally a pest from Asia. Small, soft-bodied, sap-sucking insect. Kind of cute when you see one by itself, but when you get thousands of them on a soybean plant, it's not such a good situation. They're basically sucking the life out of the plants, decreasing the yield and the quality of the soybean. And showed up first in 2000, rapidly spread throughout the soybean growing regions. We getting there, Rob?
We're pretty close. But close only counts in horseshoes and hand grenades, so.
Let go now.
I don't know what's going on here.
Oh, there we go.
There we go.
All right, so here's the cute little insect, soybean aphid. But before that, soybean, why do we care? It's a pretty big crop in the U.S. 89 million acres in Minnesota. We've got almost 8 million of those. Pretty high value production. But back to the soybean aphid, it's got these piercing, sucking mouth parts, stabs into the plants, sucks the juices out. And it, through these impacts, including affecting photosynthesis. It can decrease yields by up to 40% if they're not managed. I mentioned that it's spread throughout the soybean producing regions of North America. The red there is where it was found in the first year after detection, so in 2000. And then the yellow, you can see by, was it 2009, how far it had spread. And it's pretty much been detected throughout the soybean growing regions, and it remains there. It's much more of a pest in the Midwest, particularly in Minnesota, northern Iowa, the eastern Dakotas. When I talk to my colleagues in Michigan, Ohio, Illinois, Indiana, they're having many fewer outbreaks of soybean aphids. Sometimes for research, they even have a hard time finding aphids. But fortunately for me, for job security, we still have pretty persistent and consistent infestations of soybean aphid. So managing this critter, there's been a lot of work done over the years prior to my time in this position, and we've kind of continued from there, examining these different management tactics. Some of these include, up at the top, pest-resistant varieties. So this is through traditional breeding, so not transgenics, where we've been able to find genes that the soybean plants can have that confer resistance to soybean aphid. And you can see a susceptible leaf on the left, a resistant leaf on the right, pretty huge differences. We've also got what we call biological control. So that's when we've got predatory or parasitic insects that are working in our favor, feeding on the pests. The obvious one there is the lady beetle, the bright colored orange one, a predator. But if you look more closely, do you see the pointer here? Here's a little predatory maggot that kind of inches its way around on the leaves, feeding on aphids. And if you look really close up here, there's a little parasitic wasp. So these guys inject their eggs into the aphids. The larvae of the wasp feed inside the aphid, killing it, and then they emerge, kind of like the Aliens movie, if you remember that, where the creature pops out of the guy's chest. But right now, where we're at, management of this pest relies mainly on use of broad-spectrum insecticides. And the management recommendations for use of these insecticides rely on scouting, getting out into those soybean fields and estimating the numbers of aphids on those plants. So, imagine, acres and acres of soybean, having to traverse that field, counting aphids on plants. We can get hundreds, thousands of aphids on plants. Soybean plants can get up above waist high. If you get some windstorms, those stems get all tangled in there, and it's really not fun walking through that. And then once you get that estimate of the density of the aphids, you can relate it to a threshold, 250 aphids per plant, and that's the trigger point for lining up that insecticide application to keep the pest population from reaching a damaging level. So then coming in with what we hope is an effective insecticide to knock that population down. So we've been able to manage the pest pretty well with this approach, but there are some limitations to it. There's room for improvement. And some of these limitations are that it's time intensive and costly. It takes a lot of time to get out there and traverse those fields, estimating the soybean aphid densities. You don't cover the whole field. If you look back here, you're only actually looking at plants from a small portion of that field. So you could be missing hot spots where the aphids are actually occurring. There have been some improvements to this, speed scouting, where you're just kind of estimating how many plants have above a certain number of aphids versus below a certain number of aphids, and that's an improvement, but there's still room to improve beyond that. And kind of the bottom line is the majority of farmers want to reduce the time they're spending on scouting. They want some improved efficiencies. So for me, maybe it's laziness, like the one presenter mentioned. I spend a lot of my time out counting aphids in our research plots. Farmers, consultants are spending too much time out there. So can we take soybean aphid scouting from the ground to the air? Can we use drones, UAS, to improve scouting for soybean aphid? I am having bad luck with the technology. Yeah. There we go. So we set up a series of experiments, going back to my first PhD student working on this topic, Tavez Alves, so I showed the picture at the beginning. And kind of his initial question was, does soybean aphid affect the spectral reflectance of soybean canopies? So we're not interested in seeing the aphids themselves. These are tiny little insects. They usually occur under the leaves or down lower on the plants, so we're not expecting that we could fly over a field and see the aphids themselves. We're looking for changes, the effect of the aphids on the plants. So we started with small-scale experiments, highly controlled, where we're manipulating aphid populations to try to see what impact that would have on the spectral reflectance. And then over time, through the series of experiments that we've been performing, we're getting to more larger scale, natural, realistic, more complex scenarios. So these first experiments that Taves performed were down at Rosemont, where we set up these small caged plots. Some of the cages were infested with aphids, some of them were not. Over the growing season, we monitored those aphid populations, counting aphids on those individual plants, and then came in and got hyperspectral measures of the canopy reflectance using a handheld ground-based sensor. And here you can see aphid population growth over time in these plots from 2013 to 2014, starting out with 0 aphids per plant in July, and then by, toward the into August, we're at thousands of aphids per plant. So you can see the reproductive potential of these insects. And we were able to get some variability in aphid populations across those different plots that we had. And bringing in the spectral data, so from that broad suite of wavelengths that hyperspectral sensor was recording, Taves went in and, just for kind of proof of concept, pulled out a couple commonly used wavelengths, combined them into an index called NDVI. So we've got that plotted there over cumulative aphid days. Cumulative aphid days are a measure of aphid pressure over the season. So if you're familiar with how growing degree days accumulate over time, aphid populations accumulate over time as well. And we see a significant decreasing relationship for NDVI as aphid pressure is increasing. So that was promising. And then in the second year, we saw a very similar pattern. And these were from sample dates where we already had pretty high aphid populations. If you look at the range of values on that x-axis for cumulative aphid days, those are already pretty high. On the earlier dates, we weren't able to detect a relationship. So going beyond that, Taves did some further work with those hyperspectral data and identified some wavelengths that he found or determined would be optimal for detection of this pest, and published that paper in 2018. The previous caged work was also published. So we've got these 4 wavelengths here that looked like they were holding promise for soybean aphid detection or scouting. We took some preliminary versions of this, so it may not have been those exact wavelengths, and went to some of the folks at Centera, and they helped us create a kind of a customized sensor, multispectral sensor to put on a UAV that we use for some of the later work that I'll be talking about. So then the next question, and this is my second PhD student who's been working on this, where he comes in, and so Taves showed we can detect soybean aphid stress from a handheld ground-based sensor, but can we still detect that from the air? So if we put a sensor on a UAV, can we still detect the changes that the aphids are causing to that soybean canopy? So a very similar experimental design, slightly larger plots this time. And this experiment was conducted in southeastern Minnesota at Rosemont, and then also in Iowa over 2 different years. Very similar treatments. Some of the cages were infested with aphids. Some of them were left uninfested, created that gradient of aphid pressure over the growing season. And then, again, forced an army of undergrads to count many aphids on plants over the growing season. And Zach went out and flew a UAV with that multispectral sensor that I mentioned over these plots. He had the RGB, a couple NIR wavelengths there, and then a narrowband red. And in addition to that, we also got the hyperspectral ground-based data as well. So kind of a lot going on here. I probably should have animated it, but bear with me. So these graphs relate to the aerial data, so from the UAV-based sensor. NIR reflectance here. And we've got on the top 2 graphs are from two dates in 2017 at Rosemont, Minnesota. And then the middle two graphs, going from top to the bottom, are from the same location, but in 2018. And then we get down to the bottom two graphs, and those are from Iowa in 2018. And we see a fairly consistent trend again, where as that aphid pressure is increasing, those cumulative aphid days are increasing, we're seeing decreasing measures of NIR. For that bottom left one, which is an earlier date in Iowa, we don't see a significant relationship there, and that's likely due to the fact that those aphid populations just weren't very large by that time. But by the time we got a little later into August, aphid populations have built up more, and we see that decreasing relationship. So with this, we're able to confirm that we can still detect those changes that the aphids are causing to the soybean canopies, even when the sensor's up in the air. However, again, those were small plots that were caged, so we're using cages to manipulate the aphids. There is the potential for cages to affect plant growth, so we wanted to try to do some work in open field plots to see if we could still detect these changes that the aphids are causing. However, open fields, we can't just put the beans out there and hope that aphids will show up in high numbers in some areas and low numbers in other areas. They never cooperate. So in this study, we went in with insecticides to manipulate the aphids, and we had done some previous work to see if insecticides themselves, without the pests there, if they would affect the spectral reflectance of the canopies, and some insecticides did, some didn't. So we focused on some of the insecticides that we didn't think would affect the spectral reflectance of the canopy. And again, in this study, we did a lot of aphid counting, so it's more of a grid-based sampling scheme, stratified the field and randomly selected plants. So that's what that square on the right with all the dots represents, is one of the sample dates where we collected or where we looked at plants and counted aphids, and then we got the UAV-based multispectral data again. And here we have two different plots. So at Rosemont in the same year, we had two different fields that were set up. Plot 1 is up at the top, plot 2 is down at the bottom, and these are about half-hectare plots. And in both these, once aphid numbers got high enough, we were again detecting that decreasing relationship with near-infrared reflectance as, in this case, number of aphids per plant decreases. We weren't able to calculate cumulative aphid days here because we didn't have the season-long monitoring of aphids. It was just a couple sample dates. But even looking at just numbers of aphids per plant, we're still able to pick up that relationship. And this is a paper that Zach has in press right now. Hopefully it'll be coming out soon. And kind of the next step, where I was talking about some of my skepticism about use of UAVs or remote sensing for soybean aphid management. This was what really started changing my mind. Zach took some of this work way beyond what I expected, and in all honesty, kind of way beyond some of my understanding too as to how he made it happen. But like I said, he talked to some other people that are a lot smarter than me, like David Mulla, Joe Knight, to perform some of these analyses. So he took those data from Tavez's experiments in 2013, 2014, his experiments from 2017, 2018, pooled those together. So we had lots of data where we have these cage plots where we manipulated aphid densities on soybean. And from there, he took the hyperspectral data, pulled out those bands that Tavez identified as being most responsive to soybean aphid, and used some machine learning to try to classify the aphid populations in those plots as being either above the threshold or below the threshold. So remember earlier on when I was talking about soybean aphid management, we've got that threshold of 250 aphids per plant being that trigger point. So that seemed like a relevant value to use to try to discriminate those two populations, something that would be of relevance to crop consultants, farmers. So we used linear support vector machines to do this classification. Definitely some benefits. There are pros and cons of all these different types of machine learning. Here are the strengths and why he gravitated toward this approach. I'm not going to go into those details because I'm really not the expert on all of that. But with all that pooled data, he then separated it out, which plots had populations below 250 aphids per plant, which plots had populations above 250 aphids per plant. From those, so all the data, split it for these kinds of analyses. You need the training and testing, so split it out 60%, 40%, and then another split below that for cross-validation. And lots of machine learning magic happened and spits out output like this. So what we've got here, the gray columns are different combinations of wavelengths. So either just one wavelength alone, 2, 3, or 4. And I pushed Zach to take this approach with the thought in mind of trying to develop cost-effective multispectral sensors, right? That was brought up in an earlier presentation too. Probably the fewer wavelengths or sensors the fewer wavelengths you're trying to measure would probably be a cheaper sensor. So we wanted to try to find that balance, you know, how low can we go, but still having decent accuracy when we're trying to categorize these aphid infestations. So the highlighted areas here are some of the key measures that were coming back from these analyses. And we see in that second column, which was the optimized model, accuracy was at about 89%, significantly greater than the no information rate. Sensitivity, specificity, so true positives, true negatives, were shaking out very well. So for this work, you know, when we're looking at just soybean aphid being kind of the only factor varying among these plots, it seems like we can get by with just these two wavelengths in a sensor. Zach also did some follow-up work, which I didn't include here because I figured it would put me way over on time, where we incorporated a soybean disease sudden death syndrome. So we had aphid-infested plots, diseased plots, the combination package with the aphid and the disease, and uninfested, and trying to see how that would essentially throw things off. You know, could we still detect soybean aphid effectively, or categorize it effectively as above threshold, below threshold, when you have the disease mixed in? So in summary, we were able to show that aphid-induced stress can be detected from ground-based sensors. We were able to find some optimal wavelengths for identifying this stress. They're able to show that this aphid-induced stress could still be detected from the air, from a UAV. We're optimistic that, you know, some of these satellite-based sensors could hopefully detect the stress as well, but further work needs to be done there. And we were able to use spectral data and machine learning to classify infestations in terms of, you know, of a relevant applied value, that treatment threshold at 250 aphids per plant. So I think we're moving in the direction where hopefully, you know, maybe someday I won't have to be out in the field counting hundreds and thousands of aphids, and I can just have a technician or student fly those plots, save me lots of time and save me lots of person hours on my grant budgets. So kind of getting at why do we really care about this? My hope is that if we can make scouting for this pest or other pests more efficient, we'll get more growers, more consultants, making those treatment decisions based on some measure of what's going on with those pests in the field. We know that just because there might be a couple pests in the field, you probably don't need to treat it, right? Those plants can tolerate that. So trying to get to more judicious use of the insecticides, getting away from prophylactic applications. And if we can do that, hopefully we can decrease some of the costs of that insecticide use. So obviously, that insecticide cost, there in an earlier presentation talking about with Canada thistle and those herbicide application costs, you got the same thing going for insecticide applications. It's an input cost for the farmers, and their budgets are really tight right now, so they don't need or want those potentially unnecessary input costs. We can step back and think about some other costs that could be associated with insecticide applications, and some other students in my lab have been looking at insecticide resistance. We've been spraying these soybean aphids a lot over the last almost 2 decades, and sure enough, they've developed resistance to the insecticides, some of the insecticides. And then there are the environmental costs, right? So there's increasing concern about these insecticides showing up in surface waters within the state. Several years ago, the Commissioner of Agriculture from the state level declared chlorpyrifos, which is one of the commonly used organophosphate insecticides, declared that as a surface water contaminant of concern. And what that means is that best management practices were developed. hopefully those detections will decrease in intensity and in frequency. If they don't, there's concern that there could be regulatory implications where that regulatory entity could start revoking some of the uses of that insecticide. And if that were to happen, you know, that would really tie the growers' hands because they just don't have too many chemical tools available for management of this pest. And then potential impacts on non-target organisms, right? We hear a lot about pollinators, you know, especially the bees and susceptibility to insecticides. Eric, who's in the room here, has done a lot of work with skippers, and you know, he's got some data looking at residues of some of these insecticides showing up in these huge native remnant prairies or restored prairies, and we've done some work together looking at susceptibility of some of the skippers or surrogate species to these insecticides. So, you know, if we can improve the efficiency of scouting, I think hopefully we can start getting some collateral benefits, you know, by decreasing some of these other costs as well. Kind of where we're going from here. Doing more kind of whole field testing. You know, right now it's still at kind of the small cage study or these open field plots that are still only, you know, like half a hectare in size. You know, we really need to get to the level where we're looking at, you know, seeing if we can manage whole fields, you know, across a farm with this technology. Trying to expand to look at other potential stressors, right? There's lots of things that can affect the health of soybean. Could be drought, could be the disease like I mentioned. Through the work we're doing with the center, we're looking at defoliating insects. So insects that are chewing holes in the leaves, can that affect our ability to detect soybean aphid? Advancing this technology. So Demaz is involved with that grant as well, and he may be talking about some of that work later, but, you know, really looking at, you know, can we improve the geopositioning technology that's integrated into these sensors. And then finally, we've got some work going on with the applied economists trying to look at the economics of this, right? You know, do we really get, or can we really get an economic benefit from using drone-based scouting versus the traditional scouting? Okay. Well, maybe I can take some questions and we'll see if this catches up with my acknowledgement slides. There are lots of other people that are involved. There it is. So you can see, you know, there was earlier talk about bringing in lots of people from lots of disciplines, and that's definitely what we've been doing here. Plant pathologists, economists, remote sensing gurus, aerospace engineers, colleagues from Iowa State as well, and thankful to the various funding sources for this work that's previous and current. Thanks.
We do have time for just a few questions.
Yep.
Under a severe aphid stress, there'd be like a sooty mold on the leaf. Do you think the reflectance that's causing some of the reflectance changes?
Right, so the soybean aphids excrete a sugary substance, right, because they're living on like a Gatorade kind of diet, right? Water and lots of sugars. They can't handle all those sugars. They excrete those to kind of concentrate the nitrogen. And those sugary substances that they're excreting facilitates the growth of a fungus on the leaves that gets dark colored sooty mold, it's called. And in Tavez's experiments, 2013, 2014, he excluded data sets that had that. So we're still seeing that response without sooty mold being there. I think Zach included that in some of his data sets. I'd have to double check. I know there were multiple iterations of his analyses. But I think, yes, it definitely will affect that, that reflectance. Yeah, but that's not the only thing driving that change in reflectance. Todd?
Yeah, thanks. Very interesting. So I've got a question about soybean aphid in general and how they manifest in a field. Is it large areas and even gradients as they sort of progress over a field, or are there hot spots? And I'm kind of thinking about how you would, from a local or drone sampling technique, is it important to cover the entire field? Can you do statistical sampling on even grids, and different techniques that we use for other applications might help?
Yep, so kind of that spatial heterogeneity of the aphid infestations. And
right.
There's a student who was working on her PhD when I was doing my work here, then Rob was on her committee. She did some work looking at that spatial variability within fields, and early in the season, you get some hot spots developing where you get real high numbers of aphids on plants in some smaller areas, but those aphids very quickly redistribute themselves throughout the field. So I think throughout most of the season, it's a fairly uniform distribution across the field. Some other aphid species show a lot more aggregation. So like in potato production, green peach aphid and some of those other aphids really aggregate on the edges of the fields where you get the green canopy coming up to the brown soil, or if you've got gaps in the field, you know, where there's a skip in the planting or something, you'll get the aphids aggregating around that. Soybean aphid doesn't show as strong of aggregations there. However, talking to some crop consultants and extension folks up in northwestern Minnesota where fields are a lot larger, you know, I think they suggest that they're seeing some more spatial heterogeneity there. You know, so right now I've mainly been looking at this as a way to increase the efficiency for whole field management of soybean aphid, right? But there may be some potential depending on field size or scale where maybe we could get more toward true precision agriculture, right? Where we could just maybe spray out hot spots, or someone could come in with their flamethrower and burn out certain spots of the field or something. So not so much with soybean aphid, but some other insects have a lot more potential there. Yeah. Okay.
Okay, so at this point in the program, we're going to transition just a little bit. And instead of having formal presentations, we actually want to have a bit of free-flowing discussion led by some noted experts. And to lead that discussion, I'm going to turn the floor over to Dr. Demoz Jebra-Xabier. I'll get this right eventually, to lead our discussion. So Demoz is a professor and director of graduate studies and the director of the Minnesota Space Grant Consortium. He's in the Department of Aerospace Engineering and Mechanics. His area of research is in navigation, guidance, and control of aerospace vehicles. He is a co-PI on 3 different MIT PPC funded projects. One, as you've just heard about, on accurate detection and integrated pest management of soybean aphid, accurate detection and integrated treatment of oak wilt, and management strategies for invasive spotted wing Drosophila. So we keep Demoz very busy, and I will have Demoz introduce the panelists.
Let me set up here. Actually, I'm going to use my own computer, so if I can look up to it quickly here. Where's the VGA? That's one. The presentations are on this computer. Is that going to be a problem?
Will be captured on the
Online stuff. So, okay, what do we do? Can I get a copy? They're right here, right now. Okay. Do that.
And then you just put more microphones across.
Please go ahead. That might be the quicker way of doing things then.
So technology seems to be the bane of our existence for most of the day. So if the panelists would go ahead and have a seat, I'll go ahead and do some introductions.